Welcome to the Emery Medical Genetics Review series. In this series, we make the concepts from Emery Medical Genetics 16th edition, which is the latest edition, clear and connected. Think of it as a roadmap. Instead of reading chapter by chapter, we bring together discussions scattered across the book into one integrated concept, so you see the whole picture at once. These videos are designed for biology undergraduates preparing for advanced studies, medical students mastering their core reference, and graduates or professionals refreshing their knowledge. You can use this series in different ways. Watch first for an overview, learn alongside the textbook, or return later for quick review. Let's dive into today's concept. Hello everybody and welcome back to Instant Review. Today we want to talk about how our cells control which genes are turned on and off. So we want to speak about gene expression. This is a big deal because not every cell needs the same proteins all the time. And we know that every cell in our body has basically the same DNA material, but they do very different jobs. For example, a nerve cell sends signals or a red blood cell carries oxygen. But how does this happen? It all comes down to which genes are turned on and which are off. Cells have two main types of genes, housekeeping genes and specialized genes. Housekeeping genes are those that are always on in every cells because the cell can't live without them, like ribosomal proteins or chromosomal proteins or cytoskeleton components. But specialized genes are those that are only active at certain times or in certain tissues. For example, hemoglobin genes are only on in red blood cells, or insulin genes are only in the pancreas cells. So, this selective gene expression is what allows cells to be different from each other. But where can regulation happen? Gene expression can be controlled at many stages. Transcription, when DNA is copied into RNA. RNA processing, that's when the RNA gets spliced, capped, and given a poly A tail. Translation, which is when the RNA is turned into a protein, and post-translation, which is when the cell decides how long that protein lasts or whether to destroy it. The classic and most important level is transcriptional control. Because this type of regulation is one of the most important and classic levels of gene expression regulation. Because at this stage, the cell decides whether a gene should be turned on or off. Or in other words, the cell first checks whether transcription of this particular gene should be happen at all. This decision that transcription of that particular gene should be happen or not is made by sections of DNA called regulatory sequences or response elements. These regions can act like molecular switches, and some proteins called transcription factors can bind to these sections and determine whether the gene should be transcribed or not. Each transcription factor that are basically proteins has two main parts. A DNA binding domain that binds to specific parts of DNA and recognize that target genes, and a transcriptional activation domain, which can activate the process of making RNA after binding to DNA. So, transcription factors, besides those DNA regulating sequences, are very important players of transcription regulation because they can determine how much RNA and ultimately how much protein should be produced. However, we have other important elements and in transcription regulation. For example, we have some internal or external signals such as hormones or messenger molecules that determine transcription factors are able to bind to gene promoter. We have talked about a very important term here that was gene promoter. Promoters are sections of DNA that are located near the gene start point that initiates transcription. and they act like a master switch which can control the start of the transcription process. And they are the binding site for transcription factors. And promoters can determine when and how strongly the gene is activated. We have different types of promoters, and each have different function in gene regulation. One well-known type of gene promoters is the Tata box promoter, which is located about 25 nucleotides upstream of the start of a gene. Tata box promoter plays a key role in initiating the basal or basic level of transcription, even without external signal. So this region allows the cell to start transcribing the gene. And this is very important because if a mutation occurs in this region, so RNA production may not happen at all, or may not happen correctly. Another type of gene promoter is the GC-rich promoter. This type of promoter is located approximately 80 nucleotides upstream of the gene of interest. These types of promoters do not directly initiate transcription like Tata box promoter. Can enhance the transcription process by increasing the efficiency and binding strength of transcription factors. Or, in other words, GC-rich promoters can increase the quality and intensity of RNA production, and so they can have an amplifying role. In addition to promoters themselves, we have other regulatory elements that are important in the precise regulation of the gene. For example, enhancers, silencers, and boundary elements or insulators. Enhancers can act like volume buttons, which means that they can amplify gene activity, and so they can cause the gene to produce more RNA. But silencers, they can act in the opposite way, and they can turn off the gene or reduce its activity. And in addition, we have boundary elements or insulators, which can act like protective walls, which means that they prevent the activating or silencing signals of one gene from affecting neighboring genes. So, finally, all of these elements, which are promoters, enhancers, silencers, boundary elements, and transcription factors, can work together to form a network that determines when genes are turned on and how much RNA is produced and how the cell responds to environmental signals. So, this precise coordination is the basis for the activity of cells and their ability to respond to environmental changes. But gene regulation doesn't stop just at the first level, which is transcription. Cells can also regulate gene expression even after the RNA is made. So, this post-transcriptional control can be happen at multiple levels. For example, how the RNA is processed, how it's transported, how long it can sticks around before being destroyed, and even how efficiently it gets translated into protein. So basically, post-transcriptional regulation is like quality control. Even after RNA is made, the cell can decide exactly how much protein can be produced from that RNA. Another type of gene expression regulation is RNA-mediated control of gene expression. This is a very funny way of controlling gene expression in the cell because the cell use RNA itself to control gene expression. It's only recently that we have really understood how important this process is. There are two main players in this process, which are siRNAs and miRNAs. siRNAs, or small interfering RNAs, are super short double stranded RNAs, which are about 21 to 23 nucleotides long, and they can find their matching mRNA targets and trigger their destruction. Another type of RNA-mediated silencing can be conducted by miRNAs or microRNAs. MicroRNAs also can bind to specific mRNAs, but the differences between microRNAs and siRNAs is that miRNAs don't always destroy the mRNA molecule. Sometimes miRNAs can cut the mRNA, and other times they just block the mRNA from being translated into protein. So, RNA mediated control is the cell's way of regulating and controlling gene expression after transcription. And so, through this process, the cell can make sure the right amount of protein is made at the right time. And finally, we have alternative isoforms and splicing as a mechanism of cell to control gene expression. This is actually about just having 20,000 of genes, but producing hundreds of thousands of different proteins. And this is really interesting because we just have a limited number of genes, but very wide variety of different proteins at the same time. So, how actually is that possible? The reason behind this fact is that our genes are extremely flexible, which means that we don't need a unique blueprint for every single protein. We just have a few main blueprints and a bunch of ways to edit them. So, the main process involved here is alternative splicing. When which means when a cell copies a gene, it first creates a long and raw RNA transcript. This transcript contains all the protein coding sections, which is called exons. But before this RNA can leave the nucleus and make protein, the cell's machinery acts like a sophisticated editor, which means that the cell can selectively cut out and discards certain exons and splices together only the ones it wants. And then from that single raw draft RNA, the cell can assemble several different final mRNA messages. And each final message can lead to a slightly different product, which we know them as isoforms. But the cell also has other ways to control where and how much protein is made through alternative promoters and alternative polyadenylation. In alternative promoter process, some genes have multiple promoters. And as we know, the promoter is the sequence where the transcription machinery can start copying the gene. But different cell types, like a liver cell or a neuron cell, can use different promoters of the exact same gene. This slight change in the start position can totally alter the resulting protein's structure in different cell types or how its production is regulated. In the next strategy, which is alternative polyadenylation, the cell can modify the tail end of the RNA message. This poly-A tail is crucial because it can affect the RNA's stability, which means how long the mRNA molecule can hang around. And also, polyadenylation or alternative polyadenylation can affect the translation efficiency, which is how quickly the mRNA molecule is converted to protein. So, alternative polyadenylation lets the cell control the amount of protein being produced, not just the type of protein, which can be resulted through alternative parameter. Congratulations, you've made it! Every video you watch is a step forward in mastering genetics. Keep going. Your future expertise is already taking shape.